Is my screen available?
Not it. Yes, it is coming up. Yeah, please. Can you make it slideshow? 10 minutes time. So please go ahead.
Okay.
Yeah. So be stick to
Okay.
your time. You have 10 minutes time. Go ahead. Over to you.
Okay. My name is Firdosha. I'm a PhD student from CG Bhakta Institute of Biotechnology, Gujarat. And the title of my presentation is Regulatory T cells aging contributes generalized vitiligo pathogenesis. So, like any other organ system, immune system is also susceptible to age-related changes. There are studies which says that the deterioration can be caused in immune cells through aging. And there are ample emerging studies which are saying that due to immunosenescence or due to aging, there is a shift between protective.
Firdosh, sorry to interrupt you. Your voice is
Yes,
breaking.
ma'am.
Your voice is breaking. In case if you have any difficulty, maybe you can switch off your video so that your internet may get catched up. In case, and
Okay.
also your slides also are not moving. Please,
Okay.
yes,
Okay.
continue.
Okay. So basically, the title of my presentation is Regulatory T cells aging contributes generalized vitiligo pathogenesis. So, like any other organ system, immune system is also susceptible to age-related changes. There are studies which says that deterioration of immune competence can lead to shift between balance of protective and pathogenic immune responses. There are emerging studies which say that
Your slides are not moving. We are still at the.
it is on the first. It is on the first slide. I'm just giving an
Okay.
introduction. Yeah.
Okay.
So there are emerging studies which says that when there is a progressive aging of immune cells, there is significant effect on the frequencies of cells, their substrate distribution, as well as functional incompetence of Tregs. Therefore, in this study, we have tried to focus on how Treg cells basically, or the aging in Treg cells, contributes towards immune pathogenesis and vitiligo. So coming towards what is generalized vitiligo. So generalized vitiligo is an autoimmune skin depigmenting disorder which results into loss of functional melanocytes. And there are different factors which have been responsible. Cause has been clear-cut considered that this is causing vitiligo. So our group majorly focus on the autoimmune aspects of the study, where we have targeted regulatory T cells, which we have designated as Tregs. These are a subset of CD4 T cells, and they play a very important role in maintaining immune homeostasis. So in our previous studies, we have said that in generalized vitiligo patients, the Treg cells numbers was found to be compromised, both quantitatively as well as qualitatively. And there are some other studies which also says that the aging can decrease the Treg cells number, and it can also cause an impairment in their function. So in order to assess that, we have focused on the telomere length. The telomere is composed of repetitive DNA sequence, which is having a sheltering complex. So as we know that at every DNA replication cycle, there is a gradual decrease in the length of telomere. So till now, nobody has done the study which has investigated the telomere shortenings of Tregs in GV patients. So coming towards what was our objective, so main aim of our study was to find telomere length regulated T cells within the South Gujarat population of India, and we correlated this telomere length with the in vitro Treg suppressive capacity. So if you can see here, this is a methodology where we have collected 96 patients blood sample and 90 healthy controls blood sample. And from there, through magnetic separation, we isolated three types of cells: CD4, CD8 T cells, and T regulatory cells. Then we expanded them in vitro through co-culture system, and we did a BRD cell proliferation ELISA kit in order to determine whether these T regulatory cells were able to suppress the hyperactivation of CD4 and CD8 T cells, which were killing the melanocytes. And on the other hand, we were also isolated DNA from Treg cells, and we did a qPCR to find the telomere length through qPCR, and we found that relative telomere length, and we correlated the Treg suppressive capacity with the telomere length. So this is the overall finding. As you can see here, that is a demographic characteristics of our patients. We bifurcated patients on the basis of gender. We also did the age of onset and disease on the basis of disease activity. We divided patients onto two categories, vitiligo, and we did a severity index, that is VASI score, where we divided patients on the basis of three groups: mild, moderate, and severe, which is just in general information. Coming to the main findings. So what we found is that there was a reduction in relative telomere length in GV patients compared to controls, as well as the 0 to 20 years of age group. They have a more relative telomere length to be more reduced compared to the elderly patients. And same happened for the age of onset group. We also did one more analysis on the basis of gender, disease activity, severity, where we didn't find any significant difference. Coming to a correlation analysis data, we found a positive correlation between the relative telomere length and the suppressive capacity of CD4 and CD8 T cells. So overall, what we found of Tregs and positive relative telomere length. Also, the relative telomere length also correlated with the IL-10 and TGF-beta levels. Now, this IL-10 and TGF-beta levels are the cytokines which have the immune, main suppressive functions towards Treg cells, immunosuppressive capacity. So there also we found a significant difference. So coming to us, what were the key findings of the study? So the study found that there was a significant difference between a relative telomere lengths in Tregs of GV patients compared to the healthy controls. Also, the telomere length was significantly reduced in elderly GV patients compared to the younger patients, that is 0 to 20 years. Also, the early onset of GV patients showed more reduced telomere length compared to late onset group. So overall, what we can conclude here is that the Treg aging may be contributing factor in telomere shortening, and that can alter the Treg function in GV pathogenesis. Also, we conclude that the telomere shortening in Tregs may also result in Treg aging, and that can hamper the suppressive capacity of the Treg cells in GV patients. So this is the first evidence. Our paper is the first article which has shown that there is a decrease in telomere length in GV patients' Treg suppressive capacity, and that could be a possible reason for decreased Treg numbers and function in GV patients, which we have found in our previous studies as well. So this is overall the presentation. So thank you so much.
Thank you so much, Firdaus, by finishing on time.